in the time of the pandemic has propelled biological concepts such as virus, RNA, and RT-PCR into everyday conversation. What used to be usually discussed in the confines of a classroom, hospital, or laboratory are now media buzzwords and have made their way into family dinner conversations. Now that everybody is talking about mutating viruses, it is all the more important to understand a fundamental scientific concept, the central dogma of molecular biology. In this video, we will answer two important questions. Number one, what is the central dogma of molecular biology, and what does it tell us about the flow of genetic information? Number two, how can learning about the central dogma help us understand important phenomena such as the COVID-19 pandemic? We will learn about DNA, RNA, and proteins and the roles they play in the flow of genetic information. We will also see how changes in genetic information, known as mutations, can affect the resulting proteins. We will also discuss how SARS-CoV-2 is actually an exception to the central dogma, and what it means to have many variants of SARS-CoV-2, and how reverse transcription plays an important role in diagnosing COVID-19. But first, let's review what does it mean to have DNA as the genetic material. As the genetic material, the oxyribonucleic acid, or DNA, contains instructions for an organism's growth and functioning. These instructions are encoded in the sequence of nucleotides, or the building blocks of DNA. These are A for adenine, C for cytosine, G for guanine, and T for thymine. In a cell, genetic information flows from DNA to RNA to proteins. This flow of information is referred to as the central dogma of molecular biology, and it involves three processes: replication, transcription, and translation. Replication is the process in which information in DNA is used to make new DNA molecules, such as before a cell divides to produce daughter cells. That information in DNA can then be used to make a new molecule called ribonucleic acid, or RNA, in a process called transcription. RNA serves as a messenger that brings the instructions from DNA in the nucleus to the ribosomes, which are the protein factories located outside the nucleus. That is why they are called messenger RNA, or mRNA. When the mRNA reaches the ribosomes, a process called translation takes place. A ribosome is the cell part that strings together the building blocks of proteins, which are called amino acids. During translation, the information in the mRNA directs the ribosome to put together the correct sequence of amino acids in order to build a properly constructed protein. Proteins are molecules that fulfill many different tasks in an organism. Examples of proteins are the contractile proteins in our muscles, antibodies, which are defensive proteins, and hormones, which are signal proteins. If a mistake occurs in the process of building a protein, the resulting incorrect sequence or shape of the protein will prevent it from doing its particular function. Mistakes can sometimes happen in the process of DNA replication. These changes in the genetic information are known as mutations and are random and accidental. The variants of SARS-CoV-2 are results of mutation events in the course of viral replication. The different variants of SARS-CoV-2 have different characteristics, such as how easily they infect the host or how they react to the COVID-19 vaccine. The Delta variant of the SARS-CoV-2 virus, for example, is a highly contagious variant that was responsible for a worldwide surge of cases in 2021. Evidence has shown that the spike protein of the Delta variant makes it 50% more transmissible than the other variants. SARS-CoV-2 is actually an exception to the central dogma because its genetic information is stored in RNA rather than in DNA. A process called reverse transcription uses the information in RNA to make DNA, which is actually the opposite of what transcription does. RT-PCR, or reverse transcription polymerase chain reaction, is a technique used to detect the presence of SARS-CoV-2 RNA in a sample. In RT-PCR, the RNA from the patient's nose or throat swab sample is reverse transcribed into DNA. and a process called polymerase chain reaction amplifies or multiplies that DNA to detectable levels. Although there are several methods for diagnosing COVID-19, RT-PCR remains the most reliable and the most accurate in detecting a current SARS-CoV-2 infection. Correct diagnosis of COVID-19 allows the patient to isolate and prevent the further spread of the virus. So what have we learned today? We've learned that the central dogma of molecular biology tells us the flow of information from DNA to RNA to protein. We've also talked about how mutations are changes in the genetic material that can lead to changes in the structure and function of a protein. Lastly, we've learned that SARS-CoV-2 is an exception to the central dogma because of its RNA genetic material. Hi, I'm Dax Pinlac, a biology teacher from the Philippine Science High School main campus. Don't forget to like this video and subscribe to our channel for more lessons.